webinar of the Insights series. This is an endeavor by Inscopix to keep the neuroscience community vibrant, hopeful, and socially connected as we continue to tackle the COVID-19 pandemic. To those of you who have attended our previous webinars, we hope that you found them informative and valuable. To those joining us for the first time today, an especially warm welcome. Before we begin some logistics, I just wanted to mention that attendees can submit questions at any time during the speaker's presentation using the chat and Q&A boxes below on your screen. We will be collecting all the questions and doing our best to answer as many of them as possible at the end of the speaker's presentation. So please, no interruptions during the presentation. We'll collect all your questions and attempt to answer all of them after the presentation. We do have a hard stop at noon. So if there are too many questions that we are not able to get to, please know that it is only due to lack of time, and we'll certainly do our best to answer as many questions as we can. Today's webinar is a very special one for many reasons. We at Inscopix believe in supporting brain research in a wide array of animal species, from rodents to birds, and recently to non-human primates. And for each species, there is a distinct repertoire of behavior and capabilities providing unique and rich insights into the mechanistic understanding of the human brain. Today, we will be focusing on the songbird species. Our webinar speaker is a pioneer and leading investigator of learned vocal communication songbirds. It's my absolute honor to welcome Dr. Richard Mooney. Richard is a George Barth Geller Professor of Neurobiology and the Director of Graduate Studies at Duke University in Durham. The broad scientific focus of the Mooney Lab is the neurobiology of hearing and communication, with a specific focus on vocal learning, production, and perception. Richard has had an illustrious career with over 60 publications and many awards and accolades. It has also been an honor to support Richard's songbird research, the Decode Grant program that Inscopix launched in 2014 in partnership with the White House Brain Initiative. Today, Richard will discuss his group's work in songbirds to understand the neural mechanisms of vocal learning, highlighting circuits that enable birdsong learning, including the auditory memorization of vocal models, evaluation of song performance, and basal ganglia-dependent vocal exploration and reinforcement. I'd like to close by saying that it was a distinct honor and privilege for myself to visit Richard's lab a couple of years ago at Duke to see the songbirds singing and the Miniscope calcium imaging in real time as the songbirds were singing. You will be in for an absolute treat today, I can assure you. Thank you, Richard. The floor is yours.
Great, thanks, Kunal. It's real nice to be here. I want to thank everybody in the audience as well for taking time out from their busy Zoom days to Zoom with me. I'm going to talk to you about birdsong and birdsong learning and emphasize that, that it's one of the really beautiful examples in the animal world of kind of behavior that's learned by imitation. And why that's important to us is it's really the obvious basis of our culture is imitation, whether it's in our music, our art, our speech, or language. And just to emphasize a couple of things about this kind of learning, for example, from formal settings, master classes, seen where students seek out the very best in the field to learn how to hone behavior and imitation of the master. So the first is that it's often much more exciting for the student than it is for the master. Renee Fleming hardly looks excited talking or listening to her students sing. But for the student, this is really, really exciting. This is really arousing, stressful, and invigorating experience. The other is that even in the formal constraints of a master class, the student still has to know what behavior to imitate. For example, when working with Itzhak Perlman, the student isn't there to learn how to use an iPhone. She's there to learn how to master the violin and to play like Itzhak. And those are really hard features to capture in animal models. What I want to emphasize in the zebrafinch is that this is an animal that requires intimate social experience with the tutor, as well as the auditory experience of the tutor's song in order to learn normally. Now, the other major theme is that whether a student or a virtuoso, practice is essential. Practice makes perfect. Practice is a time to explore, and it's a time to refine performance in a way that allows us to hone our skills. And once we've acquired our skills, to use them in really, absolutely devastating performances. So Jimi Hendrix, who's one of my heroes, spent countless hours woodshedding on the guitar. He practiced every spare moment he could get. And then when he got on stage, he, pulled out all the stops, and he would do virtuoso playing, behind his back with his teeth, whatever. He did it all. And that difference between practice, a time for exploration, and performance, a time for exploiting one's skills, is something else that's available to us in the zebrafinch. In terms of the cultural transmission of the behavior, it all begins in an interaction like this. The adult male zebrafinch in the background, the tutor, sings. It's a one-sided conversation. The pupil, the juvenile pupil in the foreground, listens. In this case, he falls asleep. But in fact, that experience is transformative for the juvenile. If we use a spectral derivative of the sonogram, this frequency versus time plot, on the upper right of the tutor song, we can see that the adult zebrafinch produces this really beautiful sequence of notes. They're really well delineated. And if we looked over many renditions, they would be produced in almost the exact same sequence every time. In contrast, the vocalizations of a juvenile zebrafinch before tutoring are unformed. They have none of the spectrotemporal definition that one sees in the wild type adult song. But a few days of exposure of this juvenile to this tutor, and then a couple months of practice, resulted in an adult that can produce a very, very close copy of the tutor song. This is really remarkable. This is an animal that can listen, and through the experience of a good tutor model, can copy it almost exactly. This is very analogous to how we copy our speech sounds as the first step to language. Now, we know a lot about these processes. I'm going to emphasize two major phases of learning: a sensory learning phase, where the auditory memory of the tutor song is formed in the juvenile brain. This can be thought of as a form of auditory. It's really fast. Maybe a few minutes of total auditory experience over several days is sufficient to drive subsequent copying. And that gives rise to a second phase known as sensory motor learning, which is much slower. It seems to be a trial and error process where the juvenile sings highly variable songs and through auditory feedback assesses its own vocal performance and ultimately matches the song that it holds in memory. And just again, to emphasize the social aspects, sensory learning depends on the social interaction with the tutor as well as hearing a song, where sensory motor learning can ensue without any further social experience. But it's all for a purpose. It's not like the animal just learns to learn. He learns because he has to produce a stable song as an adult to attract a female. And in fact, this song is hyperstable, a point I'll emphasize a little later in my talk. And that stability is very important for its salience as a communication signal. Despite the fact it's highly stable, it still depends on auditory feedback for its maintenance. And what I'm going to talk to you about today are dopamine circuits that are important for this earliest stage of socially motivated sensory learning, another set of dopamine circuits that are important for vocal reinforcement, important to sensory motor learning, some of the forebrain circuits that provide evaluative signals used to guide the reinforcing element in the midbrain, a midbrain region known as the ventral tegmental area that should be familiar to most of you. And then in the last part of my talk, I'm going to talk about how basal ganglia circuits, which are downstream of the reinforcing element, enable vocal exploration, and how we're beginning to use both miniscope imaging and vocal analysis techniques to start to systematically relate neural variability, vocal variability in this vocal exploration process. All right, a little bit about the songbird brain. There is one, that's the first point. This is a sagittal view on the left and a flowchart diagram of the important circuitry on the right. There are many details left out of these kinds of diagrams, but just to emphasize a couple of things. There's a song production pathway involving HVC at the dorsum of the telencephalon that makes projections to the song motor nucleus RA, which in turn projects to the vocal respiratory planet in the brainstem and affects the learned control of song. This pathway is essential for singing throughout the animal's life. There's another pathway that HVC is part of that is directly homologous to corticobasal ganglia loops in the mammal. It includes a specialized region of the basal ganglia known as area X, as I'll show you later, through imaging experiments, is involved selectively in singing, not in other behaviors, and is essential to sensory motor learning and adult forms of audition-dependent plasticity that I'll talk about a little bit later in my talk. And this region receives, like the striatum of the mammal, receives a massive dopaminergic input from the ventral tegmental area. Okay. Now, a classic view of HVC is that it's a motor structure. The work by Fernando Notenbaum's group more than 4 decades ago established that bilateral HVC lesions rendered birds incapable of singing. And there's a wealth of other data collected since then. Beautiful work from Michael Long, Michael Fee, Dan Margoliouth, many others that's established a premotor role for HVC in singing and song patterning. However, there's also a series of publications, some earlier work from my group and more recent work from my former postdocs, Todd Roberts' group, that established that HVC also plays an obligatory role in sensory learning in this earliest phase of song memorization. And this is all published work. I'm not going to emphasize this earlier work, but turn to a more recent set of published results, spearheaded by my former postdoc, Masashi Tanaka, who came to my lab and commented that we know a lot about HVC's role in sensory learning. We know a lot about the functional anatomy of auditory inputs into HVC, but the zebrafinch depends intimately on social experience for learning. Where are the social cues coming from? And he noted that midbrain dopamine neurons had been shown to play a role in encoding social salience and social arousal, and a dopaminergic input from the A11 cell group, the midbrain tail of the hypothalamic A11 cell group, also known as the dorsal substantia nigra pars compacta in the mammal, or the dorsal SNC, provides input directly into HVC. And in fact, this is distinct from the midbrain input from the VTA into the basal ganglia. And Massa reasoned this might be a likely place to search for social signaling and a mechanism for helping HVC integrate social context with auditory information important to sensory learning. Testing this idea is highly challenging, requires recording in the midbrain of juvenile songbirds that have yet to interact with their tutor, and then studying what happens when they're first introduced to a tutor and that tutor starts to sing. What Massa saw was quite clear. In the presence of the live tutor, when the tutor was singing, A11 neuron firing rates increased. This was specific to contexts in which the tutor was both present and actively singing. Playback, which is an ineffective learning stimulus, didn't drive this activity. The presence of a live tutor who wasn't singing wasn't sufficient, nor was the presence of an adult female. Females call, but they don't sing. That suggests that A11 activity is specifically triggered by the coincident cues of a social, presumably visual, sighting of a tutor along with hearing the tutor's behavior. So like the masterclass, this is a way for the brain to tell that not only is there a good model here, but the model's producing a behavior that I should memorize and subsequently imitate. But testing that idea, Massa took a variety of approaches. I'm just going to highlight one here, and that was to lesion or block the dopamine signaling in HVC, in this case lesion, but he also used reversible dialysis techniques to block dopamine receptor signaling and saw something quite similar. And doing this at the onset of sensory learning, before the juvenile bird had had time to form a memory of the tutor song, then raising these birds up to adulthood, established that, in fact, this pathway was necessary for normal learning, at least at this early stage of development. One could think of this as possibly a trivial effect of disrupting motor activity in HVC, because HVC clearly does have a motor function. But in fact, if Massa waited another 10 days and made that lesion 10 days later after the juvenile had had sufficient time to form memory of the tutor song, then that bird copied perfectly well. And that led to the idea that this combination of activity from auditory input into HVC and dopamine release from these A11 terminals created a condition that could transform HVC activity and ultimately support imitation. Whereas if just one of these inputs, for example, in the presence of just song in the absence of a social cue, was activated, then no learning resulted. To directly test this idea, Massa optogenetically stimulated A11 terminals in HVC in naive juveniles and played song back through a speaker coincident with the delivery of this optogenetic stimulation. And he did this over several days of optogenetic tutoring sessions and raised these birds up into adulthood, never having experienced a live tutor. And in fact, these birds do copy from these optogenetically paired playbacks, whereas control animals in which GFP was expressed in A11 terminals or song playback was used without any laser illumination failed to copy at all. These kinds of experiments indicate that HVC is a node where social information from A11 along with auditory input into HVC is creating a condition that promotes subsequent learning. And I'm not going to talk a lot about juvenile sensory motor learning right now, but what I want to turn to is the ways in which adult singing and the residual variability in adult singing have been exploited for studying sensory motor learning. And getting back to the practice versus performance contrast that I mentioned earlier, adult zebra finches display this as well. An adult male in the presence of a female sings quite vigorously. This display is known as directed song. It's characterized by very long acoustic displays, including long strings of introductory notes. These are innate vocalizations that are followed by several motifs, these regions underlined by these black bars here, which are actually the learned vocal elements. These are the stereotyped sequences of syllables that the bird copies from the tutor. And during directed song, the male will produce these long strings of vocalizations repeatedly in one bout after the next. Now, he also sings when he's alone. He produces something called an undirected song, and there are many differences, a few of which are obvious right away. Fewer number of introductory notes, fewer number of motifs. The overall display doesn't last as long. It's not so obvious here, but in fact, the motifs are also delivered slightly more slowly. And if we looked over many, many renditions, we'd find that the variability in the undirected song was slightly greater than it is during the directed song. To our ears, these differences are trivial, but in terms of communicative salience, these differences are really astounding. In a really beautiful behavioral experiment done by Sarah Woolley and Alison Dope, they showed that a female, when played its mate's songs recorded either in the directed or the undirected context, strongly preferred the directed songs, even though the acoustic differences were, to our ears, quite subtle. This tells that the slightly accelerated, slightly more stereotyped behavior is really important. But it also says that there's residual variability in the adult song, and that residual variability produced during undirected song may be important to song maintenance. As I mentioned, in the adult animal, the animal is still using auditory feedback, much as we use auditory feedback, to maintain a stable vocal pattern. Now, just to give you an idea of how, when I say variable, how variable undirected song is, it's still highly precise from one rendition to the next. We can record 100 renditions of zebrafinch song and measure the fundamental frequency of a syllable in this motif, syllable D, which is centered around 625 hertz or so, and ask a student, in this case an MD PhD student, quite bright, very articulate, to say a sentence 100 times in a row as precisely as possible, and measure the frequency of this I sound in kite, and see that this distribution is extremely tight in the zebrafinch song. Now, in a landmark study done slightly more than a decade ago by Everin Toomer and Michael Brainerd at UCSF, they exploited this residual variability to drive a form of learning that we in the birdsong field refer to as pitch learning. We can measure the residual variability, the trial-to-trial or rendition-to-rendition variability in the fundamental frequency of a target syllable within the birdsong, this syllable shown here. And when it falls below a certain user set threshold, use online evaluation to trigger a white noise burst. And over many hundreds of trials, that will drive the bird to slowly modify the fundamental frequency of the target syllable into a region that escapes triggering of noise. We can think of this as an adaptive form of plasticity that allows the animal to sing without auditory feedback interference. It's trying to find a bandwidth that's unoccupied in the way. Now, again, many experiments, beautiful experiments from Michael Brainerd's group, Michael Fee's group, and others have established that the corticobasal ganglia circuits that are important to sensory motor learning are also important to this adult form of vocal plasticity. And that led to the question of where the reinforcing signals for this kind of learning are occurring. And in a study published by Jesse Goldberg's lab in Science in 2016, they recorded from anadromically identified VTA neurons that innervate this specialized region of the basal ganglia, area X, and showed that in a regime where this animal had been exposed to distorted feedback on half of its renditions, not in a pitch contingent manner, but just in a random 50% of trials, that VTA neurons behaved as if they were encoding reward prediction error. That is, on trials that escaped feedback distortion, phasic firing increased, whereas in trials that were accompanied by feedback distortion, phasic firing decreased. And this led to the idea that trial-by-trial variations in dopamine neuron activity could be driving vocal plasticity. Although that wasn't shown in this study, Aaron Heise, a former doctoral student in my lab, tested this idea directly by optogenetic stimulation of VTA terminals in a pitch contingent manner, using viral vectors to express channel rhodopsin and AAV and a fiber optic to stimulate their terminals in area X, and then using online pitch detection, she was able to trigger optogenetic stimulation of these terminals on trials, in this case, that exceeded a certain frequency threshold in the baseline distribution of pitch values. And over time, this kind of optogenetic stimulation could drive pitch learning very effectively. It did so gradually over a time course very similar to noise-induced pitch learning. Unlike pitch learning, optogenetic stimulation of VTA terminals positively reinforced the behavior. So the frequencies that were paired with light became more common in the animal's performance. A complementary study from Todd Roberts' group established that halorhodopsin inhibition of these terminals could negatively reinforce the behavior. So along with the data from Jesse's group, established the idea that the full dynamic range of VTA neuron activity was used to drive, both through positive and negative reinforcement, the bird's vocal behavior. So that leads to the necessary question of how is that performance being evaluated? And really, what are the upstream elements from the VTA that could be enabling this kind of learning? There, as in the mammal and the bird, there are many inputs to the VTA, but two that are prominent and that I want to emphasize here are the ventral pallidum, which is homologous to the ventral pallidum in mammals. This is a GABAergic population of neurons that innervates the VTA. And another region, which has no obvious homologue in mammals, known as AIV, but which can be thought of as an analog of higher auditory cortex in the mammal. And this region, AIV, is extremely interesting to us. In a study out of Michael Fee's group, recording from AIV neurons that project to VTA established that they have properties that are consistent with error detectors. That is, they're not particularly active during singing, nor are they active in response to playback of noise or other sounds. But when noise is superimposed during singing or played back during singing, then these neurons fire in response to the noise burst, as if they're detecting a distorted feedback signal during singing that is alerting the reinforcing elements in the VTA that something's amiss. Now, of course, how do you actually make that distinction that given inputs are actually functioning in an evaluative manner? And just to draw you in a little closer to the behavior and emphasize a feature of this behavior that's so attractive to people who do birdsong research, and that's its incredible temporal precision and rapidity. So, for example, in this motif, syllable D has a tonal onset pure tone element that lasts about 30 milliseconds. That whole syllable is about 100 milliseconds in duration. And we know from recordings made in singing birds that there's a premotor window that extends 50 milliseconds more backwards in time from that syllable onset. And from recordings in auditory cortical regions of the songbird, that auditory cortical processing latencies are on the order of 20 to 30 milliseconds. So one can think of a premotor window prior to syllable onset and an evaluative feedback-related window that starts about 20 to 30 milliseconds after syllable onset. And Matt Carney and Tim Warren, then a graduate student and a postdoc in my lab, respectively, combined pitch-contingent noise delivery to drive pitch learning with interference, specifically in different nodes of the circuit, either in the feedback window when evaluative information about performance was being processed in the brain, or in a premotor window prior to the generation of that element. And the twist is that noise is delivered in a performance contingent manner, but the interference is delivered in an inclusive way. That is, 100% of the trials that the bird sings elicit feedback interference. And the notion is that by combining these two elements, that is pitch contingent noise and feedback or premotor interference, we can probe in which time window and in which part of the brain rendition by rendition variations in activity are necessary for learning. And using this kind of approach, Matt and Tim were able to show that for regions that are afferent to the VTA, including AIV and the ventral pallidum, after establishing that normal noise-induced learning could occur, that when optogenetic interference was delivered in the feedback window, these animals failed to show any pitch learning. Whereas if the interference was delivered slightly before the onset of that target syllable, pitch learning occurred perfectly well. Now, in contrast, when they moved downstream to the output of the cortical basal ganglia circuit into this cortical analog LMAN, which via its projections to the song motor nucleus RA can drive song variability, and it does so very effectively, but also provides what we think is an efferent copy of that variability back into the basal ganglia, which is an important ingredient in corticostriatal plasticity models of song learning. When we delivered that interference in the feedback window, then learning occurred normally, whereas when it was delivered slightly before syllable onset, learning was completely blocked. Again, consistent with the idea that LMAN is generating more than motor variability. It's providing an ingredient to the learning-related machinery in the basal ganglia that is important to subsequent reinforcement. Along with Jiaxuan Qi, a current doctoral student in my group, these sorts of experiments allowed Tim and Matt to make a major distinction between premotor circuitry downstream of the reinforcing signals from the VTA that include and really begin with, in a sense, this basal ganglia region, area X, from an evaluative machinery that sits upstream of the VTA. Furthermore, through molecular phenotyping and optogenetic assisted circuit breaking, they were able to establish that the ventral pallidum and AIV exert opposite effects on song learning. That is, through GABAergic interneurons in the VTA, they are able to either exert a positively reinforcing or negatively reinforcing effect on behavior. And of course, this is a gross oversimplification of this circuit, but it begins to give us a sense of how the tasks of evaluation, reinforcement, and exploration are divided in the songbird brain. And because of the very close parallels in the organization of basal ganglia circuitry in mammals and birds, along with the close conservation of midbrain dopamine cell groups in mammals and birds, suggests this may provide insight into a more universal mechanism for learning very rapid vocal behaviors, such as those in birdsong and in speech. Now, in the last part of my talk, what I want to turn to are attempts to begin to understand how the basal ganglia enables vocal exploration. And just really to highlight the advantages that being able to image brain activity provide us, I want to talk about some experiments ongoing in the lab, led by a doctoral student, Jonah Singh Alvarado, that have used miniscopes to image basal ganglia activity in singing birds. Using a CAMK2 promoter, Jonah's been able to drive expression selectively in spiny neurons in area X of the basal ganglia. And using prism lenses, he's been able to get an en face view of this very large portion and specialized portion of the bird's basal ganglia. This is a video of imaging a population of spiny neurons in an adult bird. You can get a sense of what's going on just by watching and listening. I should add the bird is making many, many movements during this period. And if Jonah uses fiber photometry to image activity in the basal ganglia in area X in birds and relates that to movements that the bird makes, it's clear that the activity is specialized or selective for states when the bird is singing, not when it's making other movements of the head or body. That is, when velocity is high, there's very little signal. But when the bird is singing, there's very, very high signal. What Jonah noticed right away was there's also a stark contrast between activity patterns that were seen when the bird was in the so-called performance mode during undirected song, as opposed to when it was performing for a female. This will be obvious when I play these movies. That's a state where the male is singing by himself. If we introduce a female and just image again, activity is almost completely quenched. There's no difference if we have the bird sing directed song first and then undirected song. It's not a bleaching effect. This is strictly a sharp social context dependent change in basal. You can see this in individual ROIs. Very, very clear elevated activity during undirected singing, almost no discernible activity during directed song. Further, this change in variability, that is, or levels of neural activity, is directly related to vocal variability. Using the inhibitory opsin, archirhodopsin, Jonah was able to selectively silence basal ganglia neurons during singing and show that in the undirected state, the high levels of variability in pitch, these are fundamental frequency contours from a given syllable in the laser targeted region. During undirected song, they were high, but in the undirected state with the laser on, they were quite compressed and lower. And in fact, similar to the distribution measured when that same bird sang directed song to a female. So those state-dependent changes in spiny neuron activity that were revealed with imaging actually have functional implications for vocal variability. And the reason this is particularly appealing to me is it suggests that now we have a way of understanding how neural variability relates to vocal variability. So how do we go about linking these things together? The first is to note that spiny neuron activity in the basal ganglia is actually not highly stereotyped from one rendition to the next. For example, if we image a set of neurons in a bird and sort their activity by relative to the song behavior itself, we can see a nice, clear sequence of activity on a given trial. But if we use this same order to arrange activity in another rendition of song, we find that there isn't strong preservation of that original pattern. Some regions that were previously silent become active. Other regions that were active become silent. And the backbone, that temporal sequence that's so marked here in the first rendition, is not well preserved in the subsequent rendition. It's not totally random if we just shuffle trials and do a 2D correlation in these spatiotemporal activity patterns. We don't achieve something that is entirely random, but it's also not as stereotyped as if we optimize the correlations by pre-sorting them, rank ordering each trial to get the maximum 2D correlation in activity. It's something in between, as if the basal ganglia is producing variable patterns of activity that could be important to vocal exploration. Anyway, that's an idea, and that's an idea we're very actively seeking to test. But the question is, how do you do that? And first off, in addition to having a way to describe neural variability now, the question is how you relate that to vocal variability. And the power of the techniques developed from Michael Brainard's group notwithstanding, and the great insights that we've gotten from unidimensional manipulations of vocal pitch, the reality is vocalization is a very high dimensional behavior. There are many, many acoustic parameters. And during a process as complex as sensory motor learning, when the juvenile is imitating a tutor song, there are many, many different acoustic parameters that the brain is presumably tweaking to try to get a match between the birdsong vocal performance and the target. Here I want to mention a really fruitful collaboration with my collaborator John Pearson in the Department of Biostatistics at Duke, and a research associate, Jack Goffinet, who is working collaboratively between our labs. And they seized on an unsupervised learning technique, the very variational autoencoder, which was originally designed for image analysis and applied it to sonograms. And I should point you out also to some really beautiful work out of Tim Gentner's lab, led by Tim Sainberg, including this publication in Nature Communications, and also a very nice bioarchive from their group that isn't listed here, as well as one from Jack on this same topic. But what this technique allows is it allows a very high dimensional representation, like a sonogram in this case, 128 by 128 pixel display, roughly 16,000 dimensions, to be compressed via an encoder into a latent representation, typically 32-dimensional representation. Here shown 64, but we use 32-dimensional representation routinely now. A latent representation where variance is maximized across each of the dimensions. So instead of using hand-picked features like fundamental frequency or duration or amplitude, the autoencoder just learns a representation, a latent representation that optimizes the parameters in this latent space. It also involves a decoder which takes this latent representation and generates an image that ultimately, through training, through unsupervised learning, generates a very close match to the parent sonogram. These latent representations are really, really powerful. They can be projected, for example, using principal components analysis or UMAP techniques to show that birdsong is clustered, not surprisingly, but also within an individual cluster, for example, this syllable here, the individual dots correspond to individual renditions of the performance. So we now have a way of visualizing from the latent representation what the variability in performance is like. And that allows us now to start to ask the question of how neural variability in the basal ganglia correlates with vocal variability and vocal exploration. And as a first attempt to do this, what Jonah has done is taken epochs or trials of basal ganglia activity where activity patterns, the spatiotemporal activity patterns, are most correlated, and a subset where they're least correlated, with the hypothesis being those activity patterns that are most correlated should describe a dimension within or an excursion within the latent space that is more closely associated with other similar patterns of activity, as opposed to activity patterns that are less well correlated, which span a greater extent of the latent space representation. And in fact, that's exactly what he's seen in adult data of adults producing undirected song is that the more correlated patterns of activity in the basal ganglia, although different from rendition to rendition, are actually probing space, latent space, more finely, whereas less well correlated renditions are spanning a larger extent within this syllable cluster. What we don't know is whether or not the basal ganglia is doing this in a directed way. Is it searching just certain parts of space? Is it searching particular regions that provide a close match to the internal target, the memorized tutor song, and so on? Those are things we're beginning to explore. Of course, the deeper question in my mind is how we relate this kind of neural variability to sensory motor learning. And here, the variational autoencoder is also very useful, because instead of just being able to aggregate and build latent representations from adult performance, one can take large epics of juvenile learning and use the autoencoder and UMAP methods to project the whole suite of syllable renditions for a given syllable across a large span of development. And for example, for this syllable here, shown in this sort of boomerang shaped display here, we can then begin to relate, which is generated over, in this case, 10 days of sensory motor learning. We can then map renditions in an age-dependent manner and ask where they lie in this overall distribution. And in fact, what one can see is that in this is that there is a progression from one blade of the boomerang up towards the vertex over this 10-day span. Effectively, this allows us to look at any variant of a syllable and ask, what's the probability that it was produced at a certain age? And in combination with neural network modeling, Sam Brudner, a student in my lab, has taken these large data sets generated over a large expanse of sensory motor learning and generated or computed a mean trajectory through this latent space representation, and then used the decoder to generate, synthesize a representation of the performance that lies on that mean fit. And what that reveals, if we watch these movies, is both gradual and saltatory changes in syllable structure over long expanses of learning. Further, what you can see from these kinds of distributions is there are certain periods, for example, around 80 days of age, where many of the renditions are actually have acoustic features that are more typical of much later in development. And of course, that opens the obvious question of how the basal ganglia is contributing to this kind of exploration, and whether this early expression of adult-like behaviors is something that ends up being consolidated in the animal's performance through reinforcing actions of dopamine from the ventral tegmental area. That concludes what I'm going to talk to you about today. I'm very grateful for your time. I'm very grateful to Inscopix for giving me the opportunity to speak. I want to emphasize again the people who did the work, Jonah, Sam, Joshua, Masa, Tim, Aaron, Matt, Jack, and John, that I talked about today, and also the funding institutions, including the NIH, NSF, and Inscopix. So thanks very much.
Thanks so much, Dr. Mooney. Hello, everyone. I'm just popping in to moderate a Q&A session. My name is Sushmita, and I work in the marketing team at Inscopix. So, Dr. Mooney, that was a extremely fascinating talk with lots of interesting work from your group on this almost universal concept of vocal communication that is so interesting in songbirds, but is equally fundamental to biology and function of almost every other species. And I think I found it actually really interesting to learn some of the more unique aspects of songbird neurobiology in vocal learning too, such as the AIV. So thank you so much. We appreciate your talk very much. So just getting into a couple of the questions that have come in. We have one really interesting question from Fred Marbach, and it is what might be the effect that dopamine has in the HBC to facilitate the memorization of the tutor song? Is it plasticity intrinsic to the HBC to get the song patterning right, or also the actual content of the song?
Well, we know that dopamine's actions on HBC neurons are complex, but can be excitatory. I should mention that the A11 neurons co-release glutamate, so they have more than a single action. Dopamine seems to be a critical component for sensory learning, but activation of those terminals is presumably co-releasing both dopamine and glutamate. We don't see evidence that the dopamine neurons show strong or obvious temporal locking to features of the tutor singing. So it's not so clear that they're providing highly structured information about the tutor song. Instead, I think of it more as a permissive signal that potentiates auditory inputs from other sources, and particularly from a nucleus known as NIF, or the nucleus interface in the songbird, which we had previously shown as a major source of auditory input into HBC, and in collaboration with Ben Silvetsky showed an activation of that input during sensory learning but not after sensory learning, could interfere with copying. So we think it really acts in a way to facilitate or potentiate those inputs from that auditory region.
Okay, yeah, thank you. So another question is, what activity patterns are observed during interactions with non-partner birds in adults?
I'm sorry, I missed the very beginning. Could you repeat?
Sure. They're curious as to what activity patterns may be observed during the bird's interactions with fellow non-partner birds. So if there's no mating context involved.
In what part of the brain? I'm sorry.
It's not specified. So if you had any insights at all, that would be
right.
Okay, I think.
Sure. Yeah. Well, I didn't talk about it, but the A11 cell group is active in other contexts in the adult. In the juvenile, it seems to be, at least the juvenile male, it seems to be selectively activated by the presence of a singing tutor. In the adult, that same pathway seems to be active again while the animal is interacting, while the adult male is interacting with the female.
Okay.
So it could be states of high social arousal that pathway is very important, regardless of the specific context.
Right. Yeah, sounds like it. Okay, so another question is somebody's commenting that the singing activity was somewhat desynchronized from the brain activity in the calcium imaging video, with the singing preceding the calcium activity. If you have any comments on that.
Yeah, I mean, in fact, the basal ganglia activity elevates well before song onset, and synchronized. What we see is that the activity patterns vary. So a given ROI can participate in the same place in the song, drop out on some renditions. Its peak activity can shift across renditions. So it's not highly stable. And this actually looks different from the electrophysiological data that exists for medium spiny neurons that have been recorded in singing birds. Now, those data sets are small, but there may be some, we think in part, some divergence between the calcium signal and the electrical state of the cell. We think, in fact, we're seeing not just a reflection of spiking activity in spiny neurons, but probably the presence or absence of certain modulatory signals. And in fact, Jonah will be giving a talk on Friday as part of this dopamine virtual conference where he'll talk about exactly that.
Oh, cool. Okay, we'll have to look Jonah up after this. Okay, thank you so much. So another technical question that's come in is whether GCAMP7 has proven to be a superior sensor in your experience to GCAMP6 for imaging in songbirds.
Well, we've used both. The GCAMP7 seems, sufficient for what we're after. It's certainly bright, it's fast, and we've used in earlier iterations used GCAMP6. They both seem to work quite well. But for us, the real goal is to be able to do this in a cell types targeted way, and it's just a fortunate coincidence. The CAMK2 promoter selectively expresses constructs in spiny neurons in the zebrafinch. So that's given us a handle on that population.
Mm-hmm. Cool. Okay. So I believe Fred Marbach is asking a follow-up question. So we kind of have to revisit that very first question where you discussed the tutor song plasticity. So the question seems to be whether the NIV to HVC together with the dopamine does it help with learning of the temporal structure of the song or also the pitch content of the song? The comment is that because usually the HVC does not reflect song content as such.
What we know from the optogenetic pairing experiments that Masa did is that pairing song playback with optogenetic stimulation of A11 terminals resulted in copying of entire syllables, but often those syllables were not reproduced in the correct sequence or in the sequence that they were presented to the bird. So it could be that there is some temporal structure to dopamine release that's important for making sure that the sequence structure is reinforced properly. That's something we can't rule out.
Mm-hmm. Yeah. Okay, and another one coming in from Deborah Massini. She asks, can they learn with a playback and a fake, in quotes, fake tutor? Meaning, do they recognize the tutor as another male bird, or would they be tricked by a self-image on a mirror?
Yeah, so there's a whole, not to slight it by calling it a cottage industry, it's actually very important set of experiments, but there's a whole approach that involves kind of something called operant tutoring in zebrafinches, where tutoring can be elicited by putting an isolated juvenile in a box with a plastic model of an adult male zebrafinch, hand painted to look like an adult male with the orange cheek patches and bright orange beak and so on. And in that context, and if there is an operant playback method involving a key peck or a string pull to elicit the sound playback, then one can elicit copying. It's not as good as it's not as efficient as copying elicited by a live tutor, but it can be triggered. And our thinking about it is that in those sorts of operant tutoring conditions, the animal is becoming aroused. It's getting some of the cues necessary to put it in a state of arousal where this pathway is active. But that's not how they naturally learn, and what we're interested in is how they learn in nature, which is through social interaction.
That makes for completely sound rationale.
But maybe just to emphasize that point a little bit further, it's very likely that what the juvenile is cueing in on in the adult are visual cues, and it's probably we've thought about doing this, but haven't gotten around to it. But there's certainly a reasonable set of experiments would be to decompose visual features in the adult male, and these are obvious because of quite dimorphic plumage that distinguishes them from the adult female, like the orange cheek patch I mentioned and the zebra ruff up on his chest. That might be triggers for the juvenile to become aroused.
Yeah, that's really interesting to have them be visually present versus not. Yeah. Okay, so another question is, does variability in activity of the basal ganglia support its proposed role in exploration?
Well, that's what we're trying to answer. The evidence that it does is first that in a state where there's high variability in performance, there's high levels of activity in spiny neurons. And when there's low variability in directed song, there's very low level activity. What we see is that the basal ganglia is being dynamically modulated in a way that correlates with vocal variability. The second line of evidence is when we directly turn off the basal ganglia in an undirected context in a state of high variability, the song becomes less variable. So that's a causal manipulation that suggests that state-dependent changes in activity are actually driving variability. There are many, I should be clear about this, there isn't a single source of variability in generating song. There are many, both centrally and peripherally. There's a question of, neuromuscular variability and biomechanical variability, and those things can contribute as well. But based on our understanding of learning mechanisms, a major site where reinforcing signals are acting on performance is in the basal ganglia, and hence it makes sense that the basal ganglia is a place where variability and exploration is represented.
Yeah, that's helpful. Certainly think that with the continuing work that you're doing, that might become more and more clear. So I think we might be at the very last question, and I'm not sure you're going to appreciate this, but somebody is asking, Jagmeet Kanwal actually is asking if you can comment on variability in singing compared to speaking a sentence in humans.
Yeah, I do appreciate it. And any question from Jag is appreciated. The estimates of vocal precision in birdsong are on par with operatic singers. So deviations in fundamental frequency of pitch in an adult zebra finch or adult society finch is plus minus 1%, and that exceeds what we are typically achieving in repeated patterns of speaking. It's not that people can't become very precise. Professional opera singers sing with a similar control of pitch, but that's not standard in our species. We don't emphasize precision of pitch probably to the same degree that songbirds do.
Sure, yeah. Okay, so that seems like a nice bigger picture segue into ending this webinar. Thank you so much, Dr. Mooney. This was a very lively discussion. We appreciate your participation. And Kunal's already here, so I'll invite him to give some closing remarks.
Great. Thank you.
Thank you, Sushmita, and thank you, Rich. That was a fascinating talk and really, really exciting research in a very, very interesting species. And I think the last question is also really quite interesting just for us all to think about, why are songbirds, so precise in their pitch control, whereas, humans have to get to the level of being an opera singer to attain that kind of precision in their pitch. So really fun things to think about for us all. Thanks again, Rich. We have a great lineup of world-class scientists over the next few weeks in June and July. We want to continue this series. Hopefully, restrictions across the U.S. and across the world continue to ease up, and we can get back to what will be a new normal. But we plan to continue this series as we adjust to a new normal. Certainly this is expected to continue well beyond this crux of the COVID crisis that we're in. Future speakers on June 2nd, Amar Sahe of Harvard MGH will be speaking, followed by Ilana Witten of Princeton University on June 15th, Michael Brukas at the University of Washington on June 29th, and at least with respect to closing out speakers for the immediate future, July 13th, Zoe Donaldson, who works on the Prairie Vole from the University of Colorado, will be speaking. So again, in the interest of presenting a diversity of speakers and a diversity of species, we will also be highlighting work with the Miniscope and calcium imaging in the Prairie Vole with Zoe's talk on July 13th. We will be emailing the signups for these seminars soon. We'll be posting a schedule and topics of speakers and their presentations on all of our social media channels. If you find these webinars helpful, which I certainly hope you do, please do help us spread the word through social media and other means so we can reach out to wider audiences across the world. In the meantime, stay safe, stay healthy, and keep advancing science. Thank you, everyone.